to microbial growth because some of this information is more detailed. If you look at the chapter, for example, some of that information is very detailed. And it'd be appropriate if you're going to go into a real intense microbiology career. But I'd like to just give you some basic idea on how microbes grow and then how do we work with them in the lab to get them to grow. And so you've already covered some of this in lab. So there's going to be some overlap that you'll recognize with the idea of using the wonderful streak plate technique in order to get to know your microbe in terms of colony morphology. And, once you have enough experience working with microbes on Petri plates, you get to learn them pretty easily just by eye, but often you need additional context in order to figure out what you're looking at, because of course, there's many microbes that can look similar to this, but then if you say, well, it was isolated from a patient with this, with these types of symptoms, and then you see a certain colony type, you can often have a very good guess to what's going on, what that microbe might be. So when you think about cell growth with microbes, most microbes are well, there's a lot to it, but it's in the big picture idea, it's pretty straightforward. If they are going to kick into cell replication, they just basically get a little bit bigger. They double everything in their cell, make hopefully a faithful copy of their DNA, get everything else, up and running and doubled, and then they just split in two. And the fancy word or phrase for that is binary fission, right? So this cell is going to be binary fission. And so from one cell, you're left with two progeny cells, daughter cells, depends how you want to talk about it. But that's basically it. Your book goes into way more detail on this, but that's what I would like you to think about in terms of the average microbe. This is what they're doing, binary fission. Then you get a few interesting ones that aren't symmetrical. And sometimes it doesn't look quite so symmetrical. So here you have this cell that has a stalk, and it's actually going through cell replication. And then the two progeny cells look markedly different from each other. So every once in a while, you'll see that. You typically see this in microbes that are wild, living free. You don't typically see that in lab microbes, like the microbes you get to work with don't do this. We don't have that. We have the more typical microbes that you work with that will go through a more symmetrical binary fission. Okay. Well, just like we have generations in our family tree, as we think about the generations, you can see that happening with the microbes. And so we refer to a generation as going from one cell to two cells. That's a generation. Okay. We often want to know, how long does that take? That's very important for a lot of research that you do with microbes. And so we call it the generation time. How long does it take for a population to double? The nickname for that is doubling time. What's the doubling time of that microbe? Is often, like for E. coli, you can, if you know how to grow E. coli in the lab, you can double its population in 20 minutes. That's really fast. And there's other microbes out there that they've even got it down to 10 minutes. They can double their population in 10 minutes. 1 to 3 hours is more typical, like a big average, I guess you could say. Way faster than our generation times of decades. Mycobacterium is one of the slower ones. So Mycobacterium tuberculosis takes about 12 hours to double its population. Streptomyces bacteria that I work with, depending on the species or strain and how well we understand their growth requirements, doubling time of anywhere from 12 hours to 24 hours. So they're a little slower growers. Gotta have some patience. So very much driven by what are the microbes faced with in their environment. If you have a microbe that's wild and free, and has to fend for itself and doesn't have some lab technician giving everything it needs to that microbe, then they're gonna have a slower growth phase. Or they might not grow. They might hold steady and say, I don't have enough resources to go through binary theta. But if you're working with them in the lab, it's more of just getting to know the microbe, what makes them happy, what are their carbon, energy, and electron sources, do they need oxygen or not? And so you really do need to get you to know the microbe. So very often we want to grow microbes into a phase called the exponential growth phase. Exponential growth is usually, if you take an ecology class, for example, and they're showing exponential growth of the rabbit population or something, it's usually this green swooping line. You'll see that graphed out, where you have the number of rabbits, for example, plotted against time on the x-axis. So that's a logarithmic exponential curve. But if you put the same data on what we call a semi-log plot. Semi-log means the y-axis will be logarithmic. And the x-axis, the time axis, that will be arithmetic. So semi-log, half log. Half log, the other half is just normal arithmetic. If you do that, you turn a swooping line like this into a straight line. The red line is another way to plot that data if you do a semi-log plot. Microbiologists like a straight line like this because then you can pull data off of it easily. You can calculate the slope of the line, you can do some math and get some interesting information from that. So very often when we're tracking the growth of a microbe in the lab, we will plot out what's going on a semi-log plot. And for a lot of what I do, I want to know when my microbe goes into this so-called exponential growth, where if you plot it out on semi-log, it'll be a nice straight line. So here are some examples of semi-log plots, and you can see they're pulling data off of this. So they're looking at the slope of the line. They can calculate the generation time from this. So here's a handy equation. The generation time is equal to 0.301 divided by whatever the slope is of that line. And so that would tell you how long does it take for the population to double. So basically what I'm trying to show here is if you do a semi-log plot, you can get a nice straight line and you can pull some nice data off of that and do some calculations. Yeah, it's just standard. Yeah. Yep, 0.301 divided by slope. I don't know how they, frankly, I don't know how they came up with 0.301, but that's what it is. It's like, okay, I believe you. I don't want to know the math of that. So, but here, so when I'm growing the microbes, particularly the Streptomyces bacteria, one of the reasons I'm growing them is often to get them to produce antibiotics or quorum sensing chemicals. And I know that they tend to do that late in their exponential phase. And so I have to track them and figure out where they're at so that I can take samples appropriately. This is another plot on a semi-log. This is very important. You need to study this PowerPoint and know the phases going on here. This is like a standard thing that all microbiologists who take a basic micro class should know before walking out at the end of the semester. So this is a very typical growth curve. If you grow microbes in the lab in a batch culture system, right? So you have to define it. This is a batch culture system. So what that means is you put some, let's say, E. coli into a flask with, 500 mils of nutrient broth, put it on a shaker and get it shaken because E. coli likes oxygen, and you let it go. You don't add any more nutrients. You don't siphon away any waste. You just let it go. And then you track over time what's happening. And so if you grow a microbe like that in a batch culture, closed, they often say it's a closed system, because you're not going to add in anything, you're not going to take anything away. You just get it going and stand back and track its growth. You will get this shape. It might get elongated because it might take some microbes a longer time. So it might get a little elongated, but you still have this basic shape over and over and over. No matter which microbe. So what are we looking at here? The lag phase. If you pop E. coli, especially if, like we often work with E. coli that's, or microbes that are frozen. And so they're really sleepy. And all of a sudden you take them from the freezer, pop them into nutrient broth, and then they gotta wake up and they gotta go, what am I swimming in? And then they have to figure out, what do I need to get up and running to take advantage of my environment? And so that late phase, there's no actual growth of the microbe. They're not doubling or going through binary fission. They're just trying to figure out their surroundings. They're very active. Lots of gene expression going on, very active, but they're not growing yet. They're not increasing their population. So still very active, but no actual growth. And then once they figure it all out, whoop, they go into what's called log phase or exponential phase. So log or exponential, you can use either phrase, log or exponential. So that's your exponential phase when the population is right on track. They got their surroundings figured out and they are going big time and they're going through binary fission on a predictable cycle. And so you have that nice straight line in your log or exponential phase. Well, eventually though, because you're not adding in any more nutrients, you're not taking away their waste, that's going to become a problem because they're starting to swim around in their own waste and you're not feeding them anymore. So they're going to use up all their nutrients, get all these toxins around them. And so their growth starts to slow and they plateau. So now they don't have enough resources to keep growing. And then finally, the toxicity of their environment, because they have so many waste products, no food, then they enter into death phase. And they actually start to lyse and break open. And so your absorbance values go down. So very often what you're doing here is using to collect this data in real time. We'll grow them up in liquid and get them going. And then you take samples periodically. And then you use a spectrophotometer to measure the cell density, essentially, the absorbance value of that liquid culture. And so you can plot in real time what's going on, and which is important for me. I need to know in real time when they hit. I'm most interested in anywhere from mid exponential into the very beginning of stationary. That's where I need to know. So I need this happening in real time. Yes.
Like on the out of the lag phase before storing it, or like you store it at the beginning of what's great.
So normally what we try to do is grow up bacteria so they're in exponential phase. That's considered their healthiest phase, their most robust, they're very vigorous. If you catch them in exponential or log phase and then freeze them, that's the best. Yeah, so this is considered the healthiest phase of their life cycle. And so you'd want to capture that. Here's another example of a batch culture growth cycle. And they have two lines here. One is the optical density when you use the spectrophotometer to take the measurements in real time. And you can see it's the same basic curve as the last image we saw. This red line here is the viable count. This is data you collect after the fact. And most of the time, what you do is you take a sample out and you serially dilute it and you spread plates like what you've done in lab, where do you remember the magic range of colonies you're looking for to do accurate counts? 30. Yeah, 30 to 300. So that's where their the viable count data is coming from, that spread plate dilution scheme that you've done in class. And so it's after the fact, you have to wait for the microbes to grow, and then you count the colonies and do the math, and then you can plot it out. So this red line is after the fact data, unless you have a uber expensive cytometer machine that can count cells for you. And you don't have to do the spread plate, but it's incredibly expensive and very few people
have that on their lab bench. So that was batch culture. Then the kind of the opposite of that is continuous culture. And so continuous culture is the idea that you get things set up, you pop the microbes into nutrient broth, but you keep feeding in new nutrients. You keep siphoning off some of the waste products, and some of the cells come with that usually. And so you keep it in a steady state. So in the continuous culture system, a lot of microbial ecologists like to do this kind of thing, where they're like, I want to grow two microbes together and get them into exponential phase where they're the healthiest, and then I want to see how they interact with each other. Do they try to kill each other? Are they helping each other out? What's going on? And so getting a continuous culture system up and running is often used in ecology ideas, like how do these microbes interact? And so again, this is a piece of equipment that is pricey. It's come down a little bit in price, but it's still, all scientific equipment is incredibly pricey. I don't know, we need more companies out there because everything is so expensive. So the piece of machinery that they use for that, they call it a chemostat. Okay, so the chemostat is the machine that you purchase in order to get a continuous culture. Like, I just want to keep this culture in exponential phase forever if you want to. You are actually a walking chemostat, essentially. So you are a continuous culture. You take in nutrients, you get rid of waste, and you keep the microbes in your gastrointestinal tract in pretty much a continuous culture system. So you really are a very fancy chemostat. Way more priceless than the machinery you put on your bench space in the lab. So I like this image where they're showing the fancy chemostat here in figure C. So this is tens of thousands of dollars. And then the human. And then there you can do a continuous culture that's pretty low tech, but that requires a lab tech that you pay to keep it running. It's not automated. So this is all automated where you have precise control on input and output. But again, super expensive. So if you ever get rich and want to donate back to NMU, chemostat would be one of my wish list things. So what we're thinking about, oh, here, some other ones. Fancy systems, lots of money. Anyways, okay. So that turbidity idea. So when I grow my Streptomyces in batch culture systems, I take a sample and pop it into a spec tube and take a optical density measurement using a spectrophotometer. And I keep tracking that over time. So the fancy word for that, instead of cloudy, we say turbid. So if the broth starts to get cloudy, we say, oh, look at the turbidity in that culture. The trick you have to learn is that when the sample gets really turbid, you have to dilute it because it gets too unreliable at high cell concentrations. Your OD reading gets off. And so you have to dilute it. So when I was doing a lot of physiology experiments with a range of different Streptomyces we were working with in my postdoc. And so I had to get to know the microbe and figure out when is it producing quorum sensing chemicals, when is it producing antibiotics in some very precise tests. And because they grow so slow, those were weeks of coming in at 2 a.m., 4 a.m., 6 a.m., taking samples, and it was exhausting, but it got some, we were able to capture some really good data, but I always had to dilute. I tried to dilute when I was really fresh and not too tired, but basically what you do is you grow, at least with aerobic organisms like E. coli or the Streptomyces, you grow them on shakers where they're sitting on a platform, this platform, you can see a little bit better maybe on the left. And you put the test tubes or the flasks on clamps in there, and then you turn the shaker on and the platform just rotates. And you can vary the speed, and you can put a timer on it, so it'll stop automatically if you want. So many bacteria that you work with, as long as they're aerobic, they like oxygen and aren't poisoned by it. This is super typical. This is how you grow them in the lab. And again, tens of thousands of dollars for these types of shakers. This is an incubating shaker, both of these are, because they have a plexiglass top that you can put down, they're in the down position right now, so that you can raise the temperature in there. So you can incubate above room temperature. And those are really expensive. And then the platform shaker, this is one that I have in my lab. Right to the left of it is an incubating shaker. And then I have a platform shaker, which is just open to the room temperature. And then it just shakes. And right now I have it set at 250 revolutions per minute, which, as you can see, the liquid in those jars, it's really agitated. And so these are soil samples that we took, and we're hunting for Streptomyces bacteria in this. So we wanted to aerate. Part of that shaking is to aerate, put a lot of air, oxygen into the liquid. And remember, oxygen doesn't like to diffuse in liquid. So really shaking it at high speed like that really gets a lot of oxygen into the liquid. The other idea is to, shake it really fast so that you disperse the microbes off the soil particles, or try to. That's another reason why you do that. And just to keep everything nicely mixed, so the nutrients are nicely mixed as well. Some people will use, we've done this too, where you know, when you go to a jewelry store and you want your rings cleaned or jewelry cleaned, and they pop it into one of those sonic cleaners. You've done that too, where you take these jars of soil samples in sterile buffer solution and you put them into a sonicator and you hit go and it vibrates the microbes off the soil. And then we also shake them right after that. So that's like a double whammy to try to get the microbes off the soil so we can work with them. So what we're trying to do then is, you know, we'll isolate some microbes that we're interested in, and we're like, we want to get to know the microbes. So then we pop them into some broth and start growing them on those shakers and then take samples over time. And this is typical what you'd see. You start out with very clear broth, and then over time they get progressively more turbid. And so here you can see the optical density, which doesn't have any units, by the way. There's no units on this. But anyways, you can see the numbers are getting bigger and bigger with as increasing turbidity. Okay. So when you get to, especially this one, this is too turbid. You can't just take this measurement alone. You're going to have to dilute that. And so this next slide is trying to get at that idea here in figure C, where they're saying you get a little bit of a deviation on if you don't dilute. And so usually when I get to optical density, when my readings hit 0.5, that's when I start to dilute my sample. And then you just have to take the OD measurement of the diluted sample and then multiply by how much you diluted it by to get the actual number. And then you can plot it on your graph that you're creating in real time. So that was one trick that anyone who does this kind of experimentation, you need to be aware of. The other figure there, figure B on the left, is just trying to show that depending on the microbe, they can have slightly different growth curves. And some grow slower, some grow faster, just depends. So using spectrophotometers and measuring for turbidity over time is very effective. You could do biomass measurements, but that really doesn't work with bacteria. Because there's sometimes you need such a sensitive scale, and really you can't even do it at low densities. It wouldn't work. So we don't really do weighing of bacteria, but we do with fungi. I've done that many times with the fungi I've worked with, where I grow up the fungi, even if I grow them up in a liquid culture, usually their biomass gets significant enough where I can go ahead and drain away, filter off the liquid or centrifuge it away. And then you can dry the fungi, the hyphae, in an oven and take its dry weight. And that can be very effective. However, you're killing the fungi, so, you know, it's a destructive sample.
Talking about like. Culture, like the West Indies.
Yep. Yeah, you can grow them. Well, dry, if you want the, dry weight or experimental, then if you're gonna dry them. Sometimes I just take a wet weight. I don't wanna kill them, because I still wanna use them for something, you know, so you don't always have to do a dry weight. Dry weight's more precise, but wet weight, we do that often with fungi, because we still wanna work with them. So, but you need lots of cells for this, lots of biomass. So the fungi, because they're much bigger, and you can grow fungi either, you know, you can grow fungi in agar, but then that gets complicated to try to weigh them. So you don't, if you're gonna do a biomass measurement, you grow them up in liquid, and there's special, I don't have a picture of it, but there's special bottles that I purchased. They're called Roux bottles, R-O-U-X, Roux bottles. They look like giant flasks that you hide liquor in and put in your soup coat, you know? They're skinny. You know what I'm talking about? Instead of whiskey, you put fungi in there. So you put broth and the fungal mycelium in there, and then you just lay it down on its side. What that does is it creates a nice thin surface area of liquid in that bottle. And the mycelium just form this beautiful carpet of growth over that liquid. And then it's very easy. And this is amazing. You can take that fungi and that little bit of liquid, pop it into a normal blender that you have in your kitchen, blend the heck out of it, and it doesn't kill the mycelium. I remember the first time I did that, I'm like, I'm sorry, and I hit go and, you know, blending it out. I'm like, this isn't gonna work. Sure enough, you played it out, boom, they're happy. So as fungi are awesome. So you have this, it sounds like an experimental protocol. It's just gonna annihilate the hyphae and the mycelium, and they're like, yeah, whatever. I don't care. So biomass measurements, they're not extremely sensitive, like OD measurements, it's a little bit, I like those, but sometimes that can be hard to do with, especially for fungi. But you can do this dry weight or wet weight for fungi. Cyanobacteria are usually big enough because they usually form big colonies of cells, and their biomass can be significant. Cyanobacteria are just physically bigger than typical bacteria. And then of course algae too, so. Yes. Roux, R-O-U-X. I'm guessing it's named after a person who developed it, but they're big glass bottles that look like the flasks that you hide booze in. Just try to, you know. Did they pop up? Are you looking on your laptop? Does it look like that? Yeah, that I don't know. Tell I don't cook. I bake, I don't cook. It should look. Let me just look real quick. I found out about them when I was in grad school because I had to grow up a bunch of ectomycorrhizal fungi and get their biomass. Yeah, there they are. Ah. So, yeah. You see how they're on edge, they're really skinny. But when you lay this down on its flat side like this, it creates a nice thin layer. You put about 90 to 100 mils of broth in there, and it just creates this nice thin layer. Because most fungi like oxygen, so you can't like have this too full and you want enough airspace in here above the liquid so that the fungi can easily grab oxygen from the airspace and use that for growth. But then it forms this lovely carpet of mycelium growing across the bottom of the roux bottle when you lay it down. Okay, so back. Another technique that I've had a lot of history with, and not so many good ones, membrane filtration technique is a system that can work if you have good equipment and tight connections where nothing leaks, which is often a big ask. So, but it can be useful. What you do is you can grow up bacteria in liquid, and then you can filter them through a filter and catch the bacteria that they'll be trapped on the filter membrane because they won't be able to fit through it. The liquid goes through, but they're caught on the membrane. And then you can actually take the filter and transfer it on top of an agar, plate with agar and incubate. And then you can count colonies that show up on the filter. So if you grow up bacteria in the lab, you often, almost always have to dilute in order for this to work. You want individual colonies to show up. So if it's too concentrated, just gonna be this long of growth and you can't count colonies. We do this in microbial ecology where we go take samples out at lakes and rivers and ponds. And often we can just pour that right in and do this. But sometimes if it's really polluted system, we have to dilute. So what you're aiming for here is to get a sample diluted enough so that when you capture the bacteria or the microbes on the filter, they'll grow in discrete colonies that you can count, just like what you did with the spread plate technique where you counted colonies. And then you can estimate population size from that. So this is what it looks like. So I used to use these all the time where you have your support here where you put down your filter and then you plug that into a sidearm flask. And then this is just the funnel that you pour your sample into. You turn on the vacuum pump and it sucks your sample through that membrane. All the microbes are captured on the membrane, and you take forceps and peel that off and put it onto an agar plate and then just incubate. And then hopefully you'll get discrete colonies that you can count, and then you can use the math for that and try to get population sizes from that colony counting. So it would look something like this. So these, they were shooting for, these were water samples that were taken like at swimming beaches where they're assessing if they need to close the beach because of pollution. And so this would be one way to do that, where they take a sample and then run the membrane filtration technique and incubate, and then they count colonies. And we have laws on how many E. coli cells in particular can be found per mil of beach water. And if you go above that, you have to close or put out a warning not to swim. Or, well water, it's the same thing, not to drink. The well water if the counts are too high. And so this is something that's been happening with water quality analysis for decades. And so it can be very handy. So they've designed different kinds of agar now that makes it very easy for E. coli to visually look different. So why do you think they're targeting E. coli? E. coli doesn't always make you sick. Why should we care about E. coli? Model organism. So you have to follow the thought process here. If E. coli is present, chances are it came from fecal contamination. Because E. coli just doesn't float around in water naturally. It's just not there. And so if it's present in the water, chances are feces were in the water. And even though most E. coli doesn't harm you, the other stuff in the feces might. And so E. coli is just super easy to work with in the lab.
And so it's a model organism. We use it as a proxy to the more dangerous ones that might be lurking in the feces, whether it's human feces that, or geese or, other feces from some sort. And so E. coli is easy to test for, easy to grow. And then we say, well, if there's that many E. coli, chances are there's other fecal, potentially pathogens present in the water. So these are different agar recipes they've created over the decades for easily visually seeing presence of E. coli over other microbes. And so it can be pretty quick test and easy to assess. You don't have to be super well trained to do these tests. Here's another thing you can do if you want to measure how many cells are present. Look under the microscope. So now I wouldn't want to do this all day, but they do make special microscope slides that are called counting chambers. And I'll show you a picture of it. But basically, it's a microscope slide that has a grid pattern etched on it. And with some wells etched in it as well, so that you have a place to pipette your sample, and then it kind of wicks across this etched engraving portion of the slide, and it lets you count. And so you can get from that, you can count how many cells per milliliter of sample that you put in onto that slide. Problem is, unless you do some very special staining in this process, you can't tell if they're dead or alive. So if you want to know that information, this isn't the best way to do that. And usually we use fluorescent staining if we want to know if they're dead or alive, because you can stain cells that are dead. They'll come up with a special stain, definitely look different. And so you can tell that. How precise is this? Would you want to count slides all day, 8 hours a day? Ooh, the eye strain. I don't know. Staring through a microscope for longer than an hour, at least for my eyes, it's like, okay, I need a break. So, how accurate is this? That's up for debate. And then if they're modal, good luck. If they're swimming around as you're trying to count them, yeah, that's a lot of fun. So you can add what's that that liquid you put down to make protists stop swimming, slow mo. I don't know. It's this thick viscous liquid you can put down on a microscope slide. I suppose you could do that, but it's challenging. If you have a really low population size, like a really clean lake water, and you're trying to do this, it's like hunting for a needle in a haystack. It's really hard. And you could concentrate your cells, you could centrifuge your sample and then resuspend in a smaller volume. That would work, but it's a lot of extra steps. So microscope counts, I'll do it occasionally, but that's not my go-to. I tend to use spectrophotometers and do optical density, but it is something you can do. So this is the counting chamber. It's often called a hemocytometer because clinical labs use something very similar to count red blood cells. And so, but here's the etching. And then the little troughs that you pipette your sample. And then this is what you would see on the far right here. That's what you see when you look through the microscope. And so there's this grid pattern, and the counting chamber has directions that come with it, and it'll say, count so many of the bigger grids, and then they'll give you a formula that you can use to calculate total cells per volume. And so it's doable, but like I said, it's not something I do for a big, long experiment. This is more of a quick check that I'll do if I just want a snapshot of where we're at. Sometimes I'll do this. Here's dyes, fluorescent dyes that can be very useful for making certain cells, whether you can do live dead. So here the dead ones are orange, the live ones are green. So you can do those types of stains. You can stain and say, I want only bacteria to show domain bacteria versus domain archaea, or I want bacteria versus fungi, and you can stain them differently and they'll pop differently. So you can do, there's a lot of different stains to pick from, and it's pretty versatile. You have to have a special fluorescence microscope, which we have, and it's very, very handy. But if you're really going for the super duper, this is what you do. You'd use a flow cytometer. This is the one that costs a bundle of money. But you can actually then do fluorescence with it. And so these are like, this is the cream of the crop, where it's FACS is the acronym, fluorescence activated cell sorter. So not only will it count your cells, it can sort them into different little containers for you if you want. And so they have a flow cytometer at the hospital that one of our grad students asked permission if she could use it for her master's research. And they were nice enough to say, yeah, she couldn't touch the machine though. The technician had to do it all, but it was incredibly fast. They can count thousands of cells in under like 10 seconds. It's just phenomenally fast. So, and then this one can actually sort different cells that you're interested in. They use laser. Like, do they have that? They don't really show it. Oh yeah, here, there's a laser. So they use laser light and then the fluorescent tags, they can get it to, I don't know how they actually separate it. It's by charge, maybe. The dye might be charged differently. I don't know. But I've actually never seen one in action. The grad student went over herself to the hospital and watched them use it. So flow cytometer, cream of the crop. If you can do that, you can, it's amazing how fast. You did this already in lab where you did a viable count, which is the serial dilution, the spread plate, incubate the plates. So viable count. Viable means living. So you're only counting what grew up out of that sample. The viable cells were counted. So the viable count. So you did that. And spread plate dilutions, doing a dilution scheme and then spread plating that is very, very common. I mean, you see this in the published literature as a way to estimate population sizes of your sample. So as you said earlier, 30 to 300 is what we're going for. Those are plates that have good solid data to do statistics on. So the whenever you do a viable count like this, where you have to serially dilute, like we gave you a very short dilution scheme where you only had to dilute the sample a couple of times. I've had to dilute all the way out to like 10 to the minus 20 sometimes, where I have a big row of test tubes and I'm diluting and diluting and serially diluting and then plating that all out. And it's, at the very end of the dilution that I finally get countable colonies. So it really does depend on what your sample is. And the sample I'm thinking of that took such a long dilution scheme, it was worm castings. Worm poop, if you've ever popped that into some water and shaken it around and then serial dilute it, there's so many microbes in that you have to extend the dilution scheme way out in order to get discrete colonies to show up. But here you can see a fairly modest dilution scheme. And they're showing the gradual decline in numbers until you can get countable plates. And then some real samples there. TMTC, by the way, that means too numerous to count. So that's what you put in your lab notebook, TMTC. And those plates are useless, but got some countable ones there. And then here, I don't know if this, I think this is in your lab too, but this is showing you how to do the actual calculation, where if it's a 10 to the minus 3 dilution, you get 159 colonies, you just multiply the positive value. So you multiply by 10 to the 30, and then that will give you your numbers per mil of your original sample. So it's per mil of your original sample. And then depending on what you spread plated, you might see something like this. So very often with soil samples, water samples, you'll get a mixed population. So when you look at all these different colonies, everything that looks different is different. They're different species. So you can actually get a little taste of what's the diversity in that sample. Is it, if you see lots of different colors and colony shapes, you have pretty good diversity in that sample. If it all looks the same, pretty uniform, then you know that there's not the greatest diversity. With the big issue with these types of experiments that you always have to keep in mind is, what is your auger choice? What did you decide to use? How did you decide to incubate it? You have to give that a lot of thought. And then you need replicate plates. If you want to do stats, you have to do multiple plates per dilution for stats. So that can get expensive. Every once in a while, you have to concede that maybe two cells landed on almost the exact same spot on the plate, and those two cells just grew into one colony. So you're under counting if that's the case. In general, all microbiologists know that this method does underestimate counts. Also, if you take a soil sample and plate it out onto nutrient agar, do you think all the microbes in the soil are going to grow on nutrient agar? No. So that's a huge thing that you have to be aware of. You're limiting who's growing on that plate. It's only going to be microbes that like nutrient agar. Some microbes hate that and they won't grow on it. So you're getting a slice of the pie of what's truly there. The best way, if you really want to take a soil sample and say, who's in this? You send it off for DNA analysis. That's the best way that we have right now. But that's can be expensive. Especially if you say, you send it off and say, you isolate the DNA and get it down to species level. Who's here? And they can do that. They can take a mixed sample like that, extract all the DNA and figure out who's there. It's amazing, but it's not cheap. It's a lot cheaper than it was, but it's still not cheap. So DNA sequencing would be the way to go for that. And then there's just some microbes we have no clue how to get them to grow in the lab. They're just so well tuned to their native habitat that we have no clue. We haven't figured it out how to get them to grow. We're not smart enough for that. And we now recognize that because there's been enough data out there where they have taken a soil sample and they do spread plates and they look to see who grows. And then they take that same sample and send it off for DNA analysis, and there's a boatload more microbes there than what they captured on a plate. And so we know that there's many, many that we cannot get them to grow in the lab. And so we always have to keep that in mind too. All right, we'll stop there. A few more things to say about growth on Wednesday.